So this is commonly called hair cap moss, and botanically it is referred to as polytrichum. You would have studied funaria in your undergraduates, which is the moss example for moss. So here we see another type genus, polytrichum, which comes under Polytrichopsida class, Polytrichase, Polytrichaceae, Polytrichum. This is this is very common in uh, temperate regions as well as the hilly regions, where you can see them growing on the soil. It resembles uh, like a moss. It's quite big. You can see it very clearly. This is the diagrammatic representation of the polytrichum with sporophyte, along with the sporophyte. And this is the antheridial cluster of polytrichum, and you can see the archegonial leafy structure on the thallus. So it's a leafy thallus. It is the body is distinctly divided into leafy shoot and root-like structure or the rhizoids. It gives a picture of a higher plant. It may not be true root as you see in higher plants, but it shows advanced characters when compared to the hornworts and the liverwort. So the morphology itself has distinct divisions into root-like portion, stem-like with leaves arranged. So this is an advanced bryophyte, and occurrence is very common, worldwide distribution. Wherever you have cool temperature, tropical and temperate regions, you can see polytrichum growing. The main plant body is a gametophyte. It has a rhizome, which is the underground part, which bears rhizoids. Okay, and horizontal position, it grows in the horizontal morphology. It grows underground, bears few leaves also, and they have the rhizoids. They are rich in oil globules. And another part is the upright leafy shoot, you call it. Okay, and the leafy shoots are very long. You can see it with your naked eye. It arises from the rhizome. And you have a central axis. We don't refer to them as stem as such. We refer to them as central axis, and they bear leaves arranged spirally. Okay, and the leaves have broad leaf bases. They taper to the end. So the basal portion is broad and the end is tapering. They don't have actual midrib. They don't possess midrib, but you have an indication of a midrib portion. Slight distinction is there when compared to the other leafy worts, leafy liverworts. Porella, we saw they doesn't have a midrib, but here you have a distinct midrib like distinction in this leaf. So you can see a sheathing leaf base at the point where the leaf arises from the axis, main axis. You can see a leaf base covering. So they have chlorophyll, chloroplast. They are filled with chloroplast, and you can see the internal structure of the leaf. Very simple. The margin of the leaf is single celled. The margin of the leaf is single celled. The midrib portion shows some amount of thickness. So here you have a distinction as a midrib region and a margin region, which is an advanced character. So you have a lower epidermis, distinct lower epidermis is seen. Okay, and you have few cells like sclerenchymatous cells are seen in the midrib portion, and you can see thin parenchymatous cells are seen in the middle region of the leaf. Anganga, you can see sclerenchymatous cells, and each cell on the above above the midrib portion, you can see the cells are arranged vertically. Uh, Single cells are layered vertically with which are filled with chloroplasts. They are called lamellae. You can see the green lamellae, which are composed of single-celled, vertically placed, thin-walled parenchymatous cells, and they are called the lamellae. This is the main photosynthetic region of the leaf, polytrichum. The main axis or the stem, as it is referred, it also shows a distinct region. You have a firm epidermis from where you can see the rhizoids are rising. Thick cortex you can see, and a central cylinder is seen. So it is slightly advanced from the other bryophytes. So the main axis is distinctly divided into three portions. So you can see in some places the cells, the parenchymatous cells are thick walled. Okay, they actually show the thing of developing into the xylem and the phloem. So the central cylinder is composed of thick-walled dark cells with living protoplasts. Okay, thick-walled dark cells. They are called steroids. Thick-walled dark cells, which are called steroids. Okay, and thin-walled empty cells, which are called hydroids. Thin-walled empty cells, which are called hydroids. So the steroids have protoplasm. The hydroids do not have protoplasm. They are going to actually, they are the starting point in the evolution for xylem and phloem, the vascular bundles. So the primitive vascular tissues, you can call hydroids and steroids as the primitive vascular tissues. So you in the middle you have the cortex, which are having large cells, parenchyma cells, and you can see leaf traces in some when distributed in the main axis. So leaf traces are also present in the cortex. Vegetative reproduction is through fragmentation, or through the protonema portion. That is when the spore develops, spore germinates. It gives rise to a filamentous green structure, which is called protonema. And you can see buds growing in the protonema. So these buds, when they get detached from the protonema, they give rise to a new thallus. So vegetative reproduction you can refer to as vegetative reproduction. So fragmentation is also seen, where the rhizome can give rise to, when they get fragmented, they can give rise to a new thallus. Death or breaking of shoots separates the branches, and the branches behave as independent plants. They can give rise to a new. So this shows a male and a female gametophyte, dioecious. You can see male plant and female plant. So you can see the plant with a male shoot and a female shoot. Okay, so with antheridial cluster of antheridium, you can see antheridial cluster. Okay, and the Archegonium also develops as a cluster, apical cluster, which is bound by leaf-like structures called perichecium. Perichecium. So perichecium also develops along with the archegonial cluster. Similarly, antheridia are also enclosed by leaf-like structures called perichetal leaves. So sexual reproduction is again very, very distinct here. It is polytrichum is dioecious. As I told you, you have antheridial male plant and a female plant which grows nearby. So antheridial heads, you can see at the time of the development of the antheridium, you can see the apices, or the apical portion of the, they bear axillary clusters at the tips of the leafy stem. They are surrounded by a rosette of leaves. You can see the rosette of leaves, cluster of leaves, which are called the perigonial leaves. So this is the antheridium. Okay, this is the perigonial leaf. At the top, you can see this kind of picture. Perigonial leaves are spirally arranged. So antheridia are produced as a cluster or in groups. So paraphyses also occur among the antheridia. Paraphyses are hair-like, sterile hair-like structures which offer some kind of protection. So mature antheridium is club-shaped. This picture shows you the matured antheridium, which is club-shaped. The development of the antheridium is similar to what you have seen in the previous type studies. You have a short stalk with a jacket layer and a developing androcytes. Androcyte mother cells, they give rise to biflagellated anthrozoids or biflagellated sperms. So this is the development. This is the structure of the antheridium, TS of the antheridial head of polytrichum, where you can see some 5 to 6 antheridia developing. And these are the paraphyses, which I told you, sterile hair-like structures which are developed along with the antheridia. And you have the perigonium. You have this is the perigonium. So the development is similar to what you have seen. Jacket initial and the androgonial cells. Archegonial head is again flask-shaped archegonium. You can see a bone at the apices of the leafy stem. It is surrounded by the pericaceal leaves. These are the pericaceal leaves. And these leaves overlap to form closed bud-like structure. Archegonia are also found as clusters. 4 to 6 archegonia develop in a single cluster. So mature archegonium is a flask-shaped structure. It has a long stalk, multicellular stalk. It contains neck canal cells, and the neck consists of 6 vertical rows of cells. So neck gradually merges into a swollen venter. So venter contains upper venter canal cell and a lower egg. So paraphyses are not present in archegonia. So development is any apical cell develops as the archegonial initial, and it divides into 2 primary stalk cell and the archegonial mother cell, and it forms a massive stalk is very distinct here, and other divisions are similar to what you have seen in the previous types genus. So the central cell forms the upper venter canal cell and the lower venter, which has the egg. So sex organs dehisce in the presence of water. Venter canal cell and neck canal cells dissolve to form the mucilage. The mucilage exerts pressure and the neck just gets disintegrated, and the mucilaginous, which there is a path formed which leads the anthrozoids to the egg. So chemotactic attraction, you can say. One of the sperm swims down and reaches the base, and it fuses with the egg. So oospore is the first stage of sporophytic generation. So again, next stage is development of the sporogonium. The oospore divides to form the upper basal cell and the lower hypobasal cell, upper epibasal cell. The hypobasal cell forms the foot and the seta. So foot is embedded into the stalk of the archegonium. Actually, you have the stalk of the archegonium, and usually the foot is embedded in the stalk of the archegonium. So the cells of the seta are larger, and the epibasal cell, that is the upper cell of the oospore, forms the upper portion of the seta and the capsule. So epibasal cell divides to form the young embryo, and it is surrounded by what is called the cover cells or the calyptra. It is surrounded by the calyptra. So cells of the embryo divide to form the amphithecium and the endothecium, what similar to what you saw in Anthoceros. And that amphithecium cells are surrounded by uh, endothecium cells are surrounded by the amphithecium cells. The endothecium forms the middle portion of the main portion of the capsule, you call it. It forms the columella and the theca. So this is the sporogonium of polytrichum, structure of the mature sporogonium. So you can see the seta, the foot is embedded. You can see the seta and the massive capsule structure. So foot is buried in the tissue of the gametophyte. It is absorptive in function. It consists of parenchymatous cells which with dense cytoplasm. Seta is seen outside, several inches long, and it carries the capsule in the air. It is very quite long. Seta is quite long. You can see the seta. Okay, it's quite long and it carries the capsule. Capsule is the upper part. It is differentiated into apophysis. Okay, this differentiated into apophysis, theca, and the operculum. Operculum is the covering of the capsule. So capsule, you can see it has a capsule wall. Okay, columella is the mid portion. Okay, and in between the capsule wall and the columella, you can see the sporogenous layer. Okay, which has both spore mother cell, and in between the sporogenous tissue and the capsule wall, you can see trabeculae, which are filamentous structures which connect the sporogenous layer with the capsule wall. They have function with the dehiscence. Okay, and you have a small constriction here, and you have a swollen structure underneath the capsule, which is called the apophysis. Apophysis only connects the seta with the capsule. So apophysis is a part of the capsule. So capsule is divided into apophysis, theca, and operculum. Okay, so this is the seta. Okay, apophysis, theca, and operculum. So apophysis is the lower part of the capsule. Okay, it's continuous with the seta, and it forms the swollen ring-like structure. It is the main photosynthetic region of the capsule. So the cells contain chloroplast, and it is the photosynthetic region of the capsule. Theca is the middle portion of the capsule. It is four-lobed. Okay, its wall is several layered. Outermost is the epidermis. Okay, and then you can see air spaces, trabecular air spaces you can see in the middle portion of the, you have filaments of thin-walled elongated cells containing chloroplast. Trabeculae are thin-walled filamentous cells containing chloroplast. They form what is called air spaces in the theca region. So you have the outer spore sac wall. This is the spore sac. The middle portion. This is the spore sac. It has the outer wall and the inner wall. Outer spore sac wall and the inner spore sac wall. So you have in some species you have the inner trabeculae also. You have inner trabeculae just after the sporogenous layer. Adjacent to the columella, you can see the inner trabeculae also. Okay, the center is the solid columella. All the sporogenous cells are fertile. They form spores after meiotic division. Now, operculum is the uppermost part of the capsule. It is conical in shape. It is covered by calyptra. It is covered by what is called the calyptra. And the calyptra forms a hair-like structure. So polytrichum is called hair moss. Okay, and a constriction is present between the, you can see the constriction is present between the operculum and the theca. Okay, and this is a rim or the diaphragm. You can see the rim or the diaphragm. You can see tooth-like projections near the rim portion. It's called peristome tooth. You can see tooth-like projections on the rim portion. Okay, and this is called the epiphragm. This is called the epiphragm. The topmost portion is called the epiphragm. Okay, peristome is present in the form of a thick rim. It bears a large number of tooth-like projections. The epiphragm fills the space inside the thing. The inside space, this space is called the epiphragm. Okay, and the peristome teeth arise from the rim or the diaphragm. So this is the structure: spore capsule, okay, calyptra and operculum. And you can see the peristome teeth ready to disperse the spores. So cells of the epiphragm dry out. So this is the epiphragm, cells of the epiphragm, okay, this portion dries out and it separates the operculum. The calyptra layer falls off and the central tissue, except the spores, all the tissue degenerates and the spores are free to the at the central portion of the capsule and they come out through the pores. So wind dispersed, you can see they get dispersed by the wind. So the spores are yellow in color. Each spore is uninucleate. It has exosporium and a endosporium. Spore germinates under the favorable condition. The exosporium disintegrates, the outer spore wall disintegrate and the endosporium comes out and develops into a protonema, filamentous protonema. So here you can see the spore germination. This is the spore which gives out a filamentous structure called protonema and they undergo transverse division. You can see transverse division into a long filamentous structure. At some places you can see bud-like projections and these buds when they fall off, they give rise to a new thallus. So it is a kind of vegetative propagation also. So gametophyte of polytrichum, you can see the gametophyte is differentiated into two portions, prostrate and branched alga-like filamentous portion. So once the spore germinates, it germinates, grows to give rise to a prostrate, that is horizontal, and a vertical portion. So this is called the protonema. And you can see this is the protonema, this is the gametophore. You call it gametophore. The one which is growing upward is called the gametophore. The one which is growing horizontal is called the protonema. So filamentous protonema is not a permanent gametophytic thallus. It is transitory and it shows two kinds of branches, ordinary green ones and brown walled ones. Okay, rhizoids. So it forms two kinds of branches which are brown in color, which form the rhizoids. The green in color form the protonema, the main thallus, which is going to produce the main thallus. So when the presence of moisture, protonema grows and they at certain areas you can see leaf-like projections which are coming out of the protonema and you can see bud-like projections coming out of the protonema. So gametophore, it's called the gametophore. It is independent at maturity and is differentiated into the main axis stem and closely arranged leaves. You can see the leaf base surrounding the main axis and they grow to produce a cluster of leaves spirally arranged on the main axis. Okay, leaves are small and numerous with a broad and midrib and the lamina develops at the margin. Margin is called the lamina, leaf margin is called the lamina. So this is the life cycle of polytrichum, which shows a heteromorphic alternation of generation. All the bryophytes, they show heteromorphic alternation of generation, which have such a distinct sporophytic structure and a gametophytic structure. Gametophyte is a plant body is gametophyte, is haploid, develops antheridia and archegonia, and antheridia produces sperms, archegonia produces egg, and fertilization takes place in the archegonia venter portion and it grows into the sporogonium. You have a massive sporogonium here, which is produced by the oospore, the diploid oospore, and it has three parts: foot, seta, and capsule. And in the capsule, you have the spore mother cell, which is going to give rise to the haploid spores after meiotic division. And the haploid spores germinate to form the sporeling or the protonema, and which gives rise to the main plant, gametophytic plant at the end. So this is the diagrammatic graphical life cycle and the diagrammatic life cycle of polytrichum.